it's Eliza. I'm here with Katrina Derieg, our vertebrates collection manager here at NHMU. Now Katrina, we've got some interesting specimens on the table here. What can you tell us about these specimens that relate to our theme for behind the scenes this year, plagues and pestilence?
Yeah, absolutely. So we have several rodents and some bats as well, which are known to carry diseases. And so when we think of plagues, a lot of times we think of the Black Plague, the Black Death, right? And so here's one example of the rat that carried the fleas that carry bubonic plague. And so I've also got some fleas here in petri dishes that you'll be able to take a look at. And so museums are really important in discovering and studying diseases because specimens are more than just the skins and skeletal material. So we also take a lot of other samples with each of these specimens. So each specimen, in addition to having a skin and a skeleton, or being a fluid preserved specimen in a jar, they also have a tissue sample. And so those tissue samples can be used for extracting DNA, doing genetics work, and also pulling out viruses and bacteria, things that cause diseases. Another important thing that we collect with specimens is parasites. So that's ectoparasites like fleas or ticks, and endoparasites like tapeworms and nematodes. And all these things are part of what we call the extended specimen, all these different parts, all these different things that we can learn about each specimen. And these can be really important in tracking diseases. So for example, SARS-CoV-2 that's causing the current COVID-19 pandemic, we don't know exactly where it came from. We think it came from bats, but we don't know for sure. Unfortunately, there aren't really any specimens that represent the bats in the area where SARS-CoV-2 came from, and we can't really track where it originated.
You mentioned tracking parasites, viruses, bacteria. How different are those? Are they spread differently, or do we track them differently? Can you explain a little bit about that?
Yeah, that's an excellent question. So viruses, for example, are in the tissues or organs of the animals. So like SARS-CoV-2 or hantavirus is in the lung tissue, so we might study the lungs, and they are transmitted, through inhalation. Something like bacteria, like Yersinia pestis, that causes bubonic plague, is carried by the fleas that are on rodents. So the rodents themselves aren't the ones spreading that bacteria. It's the fleas on the rodents that then get on people and bite them and spread that bacteria. Parasites, there's a lot of different ways that parasites can infect the host. And so I could probably give a whole segment on how different endoparasites infect species.
So are these specimens found in Utah, from Utah? What can you tell us about them?
Yeah, a lot of them are. And so actually all of these, except for these bats here and the black rat, are native to Utah. So we have deer mice. Some of you might be familiar with deer mice, and these are the ones that carry hantavirus. We have the silver-haired bat, and those are known to carry rabies. We have Gunnison's prairie dog, which is really susceptible to plague, and the northern grasshopper mouse, which is not susceptible to plague, but they carry fleas that carry plague, and they'll share burrows with the prairie dogs, and then the prairie dogs get sick. Some of the other things I have here, I have vampire bats, which are known to carry rabies, similar to the silver-haired bats. But vampire bats do not occur in Utah. Contrary to popular belief, most bats are not vampires. They don't eat blood. There's only 3 species of vampire bats, and they're distributed in Central America.
Very interesting. Now I'm looking at some of these jars. You've got a lot of specimens in one jar. Is there a story behind that? Is there a reason to put so many in one container?
Yeah, so we like to collect a lot of individuals that are representative of a population. So these are samples of a population at large. And so if we only took one, that would be one animal representing a huge population of all these rodents on the landscape, right? And so if you were to grab any person off the street and say this person's representative of the entire population of Utah, that would be pretty inaccurate, right? And so we need a good sample size, and especially for disease screening, right? So we do a lot of testing for coronavirus, for example, before we get positives. Usually it's not even 50% positives. It's something more like 5 to 15% positive.
Mm-hmm.
And so you need a lot of testing, a lot of samples in order to actually get a hit.
So you mentioned that the Gunnison prairie dog is really susceptible to these fleas that carry these plagues and pestilence, right? How devastating for a prairie dog community are these diseases? Are they wiping out huge numbers or just a small population?
Yeah, so they can wipe out an entire colony. And so this has been a really hot topic in conservation because prairie dogs are the main food source for the black-footed ferret, which is an endangered weasel. And so there's been a lot of work done on studying prairie dog populations and how viable they are as a food source for black-footed ferrets that are being bred in captivity and then reintroduced into the wild. So it's actually really important to make sure that these prairie dogs are not getting and spreading plague.
So this Gunnison prairie dog, how likely is it that one that is carrying these fleas that carry the plague would transfer to a human? Am I gonna get the plague if I see one of these on the side of the road?
Most likely not, only if you're going and grabbing them out of their burrows, which I do not recommend doing. No, it's unlikely. There are certain hot spots throughout the Southwest, and there are a few plague cases per year, particularly in New Mexico, Arizona is where it's more common. And so sometimes it can happen if somebody's dog is out and catches a prairie dog, gets the fleas, brings those fleas into your home, and then they get on you. But it's not an incredibly common occurrence, fortunately.
Okay, that makes me feel a little better. What can you tell us about the work that you are doing with these vertebrates?
Yeah, so sort of my role here is to preserve specimens that have the broadest use possible for the research community. So that's ecology, evolution, disease research, conservation. And so something that I'm working really hard to do is to collect those samples, like I mentioned earlier, those parasites and tissue samples of different organs, so that researchers can come to the museum and say, hey, I am interested in screening for diseases. Can I have a tissue sample and extract viral RNA and discover if there's a disease in this population? And so building these resources, this infrastructure for researchers is really sort of my critical mission.
So we've looked at a lot of rodents today. Is that kind of your specialty in this department, just the small rodents, or do you branch off into anything a little bit bigger?
Yeah, so I mostly focus on rodents. That's sort of my main area of study. But I'm a mammologist, so broadly I study mammals. Another mammal group that's of interest when we're talking specifically about diseases is deer that carry ticks, that carry Lyme disease, which is another bacterial disease. And so that's one that's really important for land managers to study, is these large game species and how they might be vectors for disease.
Thank you so much for giving us your expertise on this today. I know these behind-the-scenes events are really informative for the public. So just as a wrap-up, while transmission is really rare, what can the public do to kind of protect ourselves from plagues and pestilence that we might find in the mammals of Utah?
Yeah, absolutely. So as a general rule of thumb, never handle any wild animal, observe from a distance. Something like hantavirus, which is carried by deer mice, is transmitted through breathing it in. So make sure that you wear a mask anytime you're cleaning out your shed or your garage or something like that to reduce transmission. But we must remember that all of these animals play a super important role in the ecosystem. And so our goal is not eradication for the sake of eradicating these diseases. It's really so that we can live in harmony with these wild animals and not pick up any nasty diseases from them.
Thank you so much for meeting with me today. Stay tuned for more looks at our behind the scenes plagues and pestilence events, and we hope to see you here soon.